So yeah, I'm a master's student at the University of Edinburgh, and I'll be presenting the project I conducted as my honours research thesis, which was on the use of enhanced urban flower meadows to promote bee biodiversity. So there's been abundant research over the past 40 years which documented pollinator declines, and these have been attributed generally to habitat loss, pesticide and agrochemical use, and climate change. The potential impacts of these declines cause concern, particularly considering 87% of wild plants and approximately 35% of food crops rely on insect pollination. So naturally, there have been strategies and initiatives being developed to mitigate these declines, often taking the shape of AES, agri-environmental schemes, such as flower strips and patches. An example of such flower patches exist at the Edinburgh foreshore, which are the result of a collaboration between the University of Edinburgh and Nature Scott to support local flower and pollinator communities. And I was able to work on the first year of data from these meadows, aiming to investigate how the creation of these meadows has impacted flower and bee diversity. So a preliminary study into these meadows. So the data was collected in 2021. I didn't do the data collection myself, but got to work on the data set. It was collected during 5 survey rounds in May, early July, late July, August, and September. The locations spread across 8 sites on the Edinburgh Silvernorse Cramond and Gypsybury foreshore, with each site having a sown flower meadow, wildflower meadow, and a companion plot of unimproved grassland. At each survey round, surveys of both pollinators and flowers were conducted. So this gave me data of pollinator abundance and diversity, as well as flower abundance and diversity. I decided to focus on the Apidae family more specifically, as these are key pollinators and all were identified to species, whereas other insect pollinators were identified to different levels of specification. So I created several linear models and generalized linear models assessing the effect of these flower meadows on the flower and bee diversity. Then I created models to more specifically represent the relationship between the flower increases in flower diversity and increases in the bee diversity. And finally, I separated the flowers into bumblebee favorites and others according to literature, and modeled their effects on bee diversity separately. So I found that in just their first year of existence, meadow plots had higher flower abundance and species diversity than companion plots, thus showing if the treatment is effective. And I found that the meadow plots also attracted higher bee abundance and diversity, showing not only is the treatment effective, it's effective at attracting pollinators. Furthermore, I found there is specifically an increase in bee abundance and species richness as flower abundance and species richness increases. I also found that the effect of bumblebee favored flowers on bee diversity was more important than the effect of the other flower abundance, and this particularly for bee species richness. So therefore, my project supports the idea that sowing wildflower patches can support greater local biodiversity, and these wildflower patches might also be helping the pollinators in providing nesting and hibernation habitat. But there remain many complexities that are unknown, and these meadows can help us find more information about the relationships. Thank you.
Thank you very much. The guys into the client and give you a 4-minute talk.
I've done this sort of thing myself before. The shorter the talks are, the harder they are to keep to time. Thank you very much. And our second speaker then is Savanna van Mesag, who's a PhD student at the University of Glasgow. Savanna.
Testing, 1, 2, 3. Hello? This is working? Hello? Oh, there we go. Yeah. Hi, I'm Savanna van Mesag. I'm a PhD student at the University of Glasgow, and I also have a supervisor at the University of Stirling. Very nearly finished my PhD. I've been working on plants growing on anthropogenic substrate sites. What are anthropogenic substrates, you might ask? These are materials produced by people, usually in various industrial processes. We have lots of legacy heaps of, say, steel slag, colliery spoil, paper mill sludge, and many, many more in countries such as the UK. Specifically, during my PhD, I've been studying 6 specific field sites, 3 of which are in central Scotland and 3 of which are in northwest England. And I was looking at plant communities and plant species on these sites, looking at things like biodiversity. Here I just have species richness represented in a pie chart. This is specifically Hill's number Q equals 0 as calculated in INEX, which is an R package. So it's a modified form of species richness, essentially reflecting what's present on the site. And we have basically, throughout my 6 field sites, I found a huge range of biodiversity levels, which wasn't necessarily to be expected. It's mostly in the mid-range between 10 and 20, as can be seen in the pie chart, but we had a few sites that were particularly biodiverse, really showing just how important these places are for biodiversity and for species richness. And I calculated various other indices, but this is just what I have to show to you today. This is the canonical correspondence analysis, which basically shows substrate variables that I also recorded on the sites in relation to some of the plant species present. And what I found is that the various elements and other variables here, including pH level, vanadium, phosphorus, and so on, all had significant influence on this plant species present. I just have a few highlighted here. The red crosses are species, and the white circles that you can see in the graph as well are communities. And so just to give an indication, you've got some species, as shown on the graph, which are differently influenced by different elements present on the sites. I also looked at indicator species in relation to certain minerals. Because I couldn't record minerals quantitatively on the anthropogenic substrate sites that I studied, I instead looked at it in a more qualitative manner. So what we have here, where the indicator values are 1, that's the maximum value. So that shows a very strong relationship between certain plant species and certain minerals on the anthropogenic substrate site. Because of limited sampling and because some of these minerals were not actually recorded that much, it would be particularly interesting to go to other sites with the same species and investigate these potential relationships further. So this is very much within the confines of the study, but it would be very interesting to see how this relates to other anthropogenic substrate sites. Many thanks for listening. Any questions?
Thank you, that's tremendous. It's great when young people get in ahead of you, isn't it? So our third talk then is by Connie Simon Nutbrown, and this is a researcher at Botanic Gardens here. Over to you, Connie. Welcome.
Thank you. Just check that everyone can hear me. Yes, good, excellent. Yes, I'm Connie. I'm a postdoc researcher here at the Botanic Gardens, and I'm going to try and explain a little bit about the project that I work on, which is Biodiversity Genomics Europe, and one aspect of the work that I'm doing, which is telling willows apart with DNA. And I'm going to try and do it in under 4 minutes, so buckle in. I will also say I'm going to be presenting some unpublished data, so any of the slides which have like raw data on them, maybe let's not tweet those just yet, because we're going to hope to get it out in the world soon. So what is Biodiversity Genomics Europe? And this is a big EU grant which brings together 2 main streams which encompass lots of other projects, such as the Earth Biogenome Project, Darwin Tree of Life, which some of you may have heard of, and I do some work with them as well. And mainly the overview is it looks at 2 main things: species diversity, based off of telling species apart with small amounts of DNA, which are known as DNA barcodes, and then also genetic diversity, which we're using sequencing whole genomes for. And those are like the 2 aspects of the project.
And I bridge the gap between those two things. So I'm bringing together DNA barcoding, which is using the small amounts of DNA, and whole genome sequencing, which is as much DNA as you can get, to better understand three main things, like the nature of species differences, or the genetic nature of species differences, which is a big aim, the degree to which plants can be identified to the species level using traditional barcoding loci, and then to design new ways of doing that better for species where traditional barcoding loci don't work as well. And how we're aiming to do that is to start with the most amount of data possible. So we're going to get the highest resolution whole genomes, and we're going to start with like the truth, the most data. And then we're going to look at how species where you traditionally struggle to tell them apart with DNA barcodes, how those separate using all of the data, and then try and work backwards to smaller amounts of DNA where you still have that resolution, but also it's easier to get that data and you're able to do it quickly, cost effectively, and maybe even like with a biomonitoring kind of assay. Just to quickly explain DNA barcoding, it's kind of like you take a very, very small fraction of DNA, and that is unique within species. So it's kind of like a fingerprint, and then you can compare it against like a reference of lots of different species, and then it will match up to your species. And this works very well in lots of plants and in animals, and not so well sometimes for other plants. And one of the species, one of the groups where it doesn't work so well is willows. As some of you may know, there's this paper from a few years ago, and they have this tree, and basically the takeaway from that is that all of the species are sort of all grouped in together. And to take this data a little bit further, if you look at the plastid genome, basically look at the colours of species, and they're all bunched up together with no resolution. If you take a little bit more data, oh, that should be a figure. Well, for ITS, you get a little bit better than that. If you look at the whole genome, most amount of data, you can see the colours will group together really nicely. And then from this, we've started working, taking less data. So we've got this, which is quite a lot of data still, but less. And again, you can see the colours group together nicely. We've got better resolution of species, but using less data than whole genomes. And we're going to keep taking it down so we can do this better. Yeah, those are my next steps. And hopefully you vaguely understood that in the quick time, and I'm available for questions.